Good morning, students. Today we'll discuss about the pest-resistant plants. Last time we have discussed about that Bt toxin and Bt cotton and Bt toxin. That mechanism already you have learned last class. And now today we'll learn about the RNA interference. What is actually RNA interference? That is the stopping the translation process. All of you know the translation process means which one? Now when the mRNA is converted into protein polypeptide chain, that is called translation. So in this process actually RNA interference means the blocking of mRNA, or we can say the from mRNA to protein synthesis, that translation process will be blocked here. That's what is called interference in RNA mechanism. So another approach here you can start about the another approach to develop the pest resistant crop against nematode parasite. So it is actually against which one? Nematode parasite, and that is the pest here. And we are raising actually by this process we are raising which one? Pest resistant crop. That crop is resistant against that nematode. What is the name of the nematode here? Meloidigine incognita. इनकॉग्निसी एक्चुअली यहाँ पे TIA होगा, तो मेलॉइडिगाइनी इनकॉग्निशिया दैट इज रूट्स ऑफ टोबैको में ये अटैक करते हैं. तो प्रिंसिपल ऑफ RNAi डेवलप्ड पेस्ट रेजिस्टेंस दैट इज साइलेंसिंग ऑफ ट्रांसलेशन ऑफ स्पेसिफिक mRNA. कोई स्पेसिफिक mRNA जो that mRNA which is actually producing the specific protein or life survival protein for that nematode, because that proteins are produced, that because of that one that nematode is surviving, that protein synthesis will be stopped, है ना. तो that is called the silencing the translation. That's what is called silencing translation of specific mRNA. कोई specific mRNA की जो translation हो रही है उसको silence कर देना है और उसको stop करना है. RNAi takes place in all eukaryotic organism as a method of cell defense. It is actually working as cell defense mechanism. Silencing of specific mRNA due to complementary double-strand RNA, a type of RNA molecule. This is the main mechanism here. Double-strand RNA, a type of RNA molecule to enhance or inhibit the translation of mRNA, binds to mRNA and prevent the translation. Suppose the mRNAs are producing from which one? DNA. DNA is the central dogma, all of you know that one. From DNA by transcription process mRNA, from mRNA it is processed the polypeptide chain, that is called protein synthesis, and that process is known as the translation. Okay, so if the mRNA is single stranded and if the dsRNA is introduced there, double stranded RNA is introduced there, or we can say that gene is introduced, that also we'll discuss that one, that double stranded RNA that is actually reducing or that binds to mRNA, and definitely तो mRNA single strand है और ये double strand RNA से again they are complementary strand is actually combining with that mRNA, and so that the mRNA is not now translated, because ribosome will not proceed now because mRNA already blocked हो चुके हैं, mRNA already bind हो चुके, complementary strand से mRNA क्या हो गया? Bind हो गए. The source of dsRNA or complementary RNA, virus or transposons, genetic element which replicate via an RNA intermediate. So that dsRNA, double stranded RNA or the complementary RNA that is actually binding with the mRNA, virus RNA or transposon, whatever is actually binding or silencing that translation process of specific mRNA, that genetic element which replicate via an RNA intermediate. So that is actually replicating via RNA intermediate. So that is called the transposon. So now these are the sources. Now we are coming to the point here. The method of genetically modifying crop through RNAi. ओके, सो हाउ, व्हाट इज द मेथड यार, बहुत ही इंटरेस्टिंग है, एक बार देख लो आप. तो वेक्टर, व्हाट इज द वेक्टर यूज्ड है? एग्रोबैक्टीरियम ट्यूमिफेसियंस, जेनेटिकली इंजीनियर टू इंट्रोड्यूस द निमाटोड स्पेसिफिक जीन्स. ओके, तो निमाटोड स्पेसिफिक जीन्स that is actually separated or isolated by restriction enzyme. Again, agrobacterium plasmid that is using as a vector here. So both are cut by same restriction enzyme, and the specific gene that is responsible for the production of the survival protein in nematodes that is introduced in the vector. Recombinant DNA is produced. Now recombinant DNA is introduced, which one? In the, we can say, the host plant. So nematode specific gene introduced into the host plant, that is called transgenic plant. So that when the recombinant DNA is introduced into the host plant or into the tissue of a host plant, cell of the host plant, and then it is culturing to form a transgenic plant. Okay, DNA introduced will produce both sense and antisense RNA in the host. So host is the plant here. So in that plant already we have introduced which one? The nematode specific genes, that DNA. So that is, that nematode specific DNA is introduced where? In the plant. Okay, so that plant cell will be produced which one here? And they are produced the sense and anti, because DNA double strand, and in that DNA the nematode specific gene is introduced, and when this DNA they are transcription is going on, so they will be producing which one? Sense strand or non-sense strand, and there they are complementary DS double strand RNA is produced there. Okay, so now we are coming, double strand RNA is produced there. Since we can say the sense and antisense RNA are complementary to each other, so double stranded RNA is produced. That in it is the RNA interference. RNAi means RNA interference. When nematode infests the plant introduction, of nematode's genome takes place, and presence of dsRNA silence the specific mRNA of nematode. So जब भी nematodes entering into the plant, or they are taking the actually it is tobacco plant, roots of the tobacco plant they are infesting. So when they are infest the plant, introduction of that nematode genome takes place. Which one? Double strand RNA that is taking place, and presence of this double strand RNA silence the specific mRNA of the nematode. Initiation of that means now the mRNA translation process is stopped there. Parasite will not able to survive in host which express interfering RNA. So that is called RNA interference. Transgenic plant resistant and protected from the parasite. So transgenic plant we are producing. First what we are doing? First we are separating the nematode specific gene from nematode. Okay, which are responsible for their survival, and same way we are actually separating the plasmid, and there we are by recombinant DNA technology, means by using same restriction enzyme we are introducing that nematode specific gene in plasmid. Again that plasmid containing the nematode specific gene introducing in the agrobacterium. Again agrobacterium is introducing her in the plant cell. So where in the plant cell, in the plant chromosome that is introduced, means which one? In plant DNA, the nematode specific gene is introduced by agrobacterium. Now when this plant DNA is and then transgenic plant is produced, and now inside the plant cell when this DNA is transcription is going on, both the strand of DNA containing the nematode specific gene, they are producing sense strand and antisense strand, and both the strand are complementary to each other. That's why they are producing the dsRNA, means double strand RNA. This double strand RNA, when the nematode infests the plant, this double strand RNA introduced into the plant nematode's body, and they are, as they are complementary to the mRNA of the nematode, so they will combine with the, or they are combining with the mRNA and stop the translation process. So definitely when proteins are not, means necessary proteins are not produced in the nematode body, they cannot survive. That is the main process here. So next we are coming here, this is actually the picture, and next one, application of the biotechnology in medicine, biotechnology in medicine, that is diagnose, treat and different diseases with effective recombinant therapeutic drugs. This is the first point here. Protect from dangerous disease, and through gene therapy new and healthy genes can be inserted in the replaced damaged cell. Targeted action of drugs by through study of genomics, and recombinant therapeutics do not induce unwanted immunological response as in case of similar products isolated from non-human source. 30 recombinant therapeutics approved for human use, and 12 of these marketed in India. Okay, so now this is coming about the biotechnological application, and genetically this we will discuss tomorrow. So now up to that we have discussed about the RNA interference. Okay.